Oral cavity. The oral cavity is bounded by teeth, tongue, hard palate, and soft palate. These structures make up the mouth and play a key role in the first step of digestion, which is ingestion. This is where the teeth and tongue work with salivary glands to break down food into small masses that can be swallowed, preparing them for the journey through the alimentary canal. The tongue manipulates the chewed food into small mass called a bolus, then moves it to the oropharynx. The next step are involuntary. The bolus passes through the pharynx. The epiglottis closes off the trachea and directs the bolus down the esophagus, and peristaltic waves move the bolus into the stomach. The pharynx is a cone-shaped passageway leading from the oral and nasal cavities in the head to the esophagus and larynx. The pharynx chamber serves both respiratory and digestive functions. Thick fibers of muscle and connective tissue attach the pharynx to the base of the skull and surrounding structures. Both circular and longitudinal muscles occur in the walls of the pharynx. The circular muscles from the constrictions that help push food to the esophagus and prevent air from being swallowed, while the longitudinal fibers lift the walls of the pharynx during swallowing. The uvula and the epiglottis make up the most important organs that aid essential functions within the respiratory and digestive system in mammals. Peristalsis is a sequence of wave-like muscle contraction involved in the movement of food and other liquid particles in a digestive tract to different processing organs present in the digestive system. This process originates from the esophagus when a completely chewed food is swallowed. These involve reflexive movements of the circular and longitudinal muscles, majorly in the digestive area, but sometimes in hollow tubes that exist in progressive wave-like contractions. Peristalsis waves are present in stomach, esophagus, and intestines. These waves could be shorter, longer, continuous, or continual that move within the entire length of the organs based on their location. Two factors that influence enzyme activity deserve further consideration here in the context of digestion: temperature and pH. Low temperatures result in slow reaction rates because overall kinetic energy is reduced, since particles are moving more slowly. Substrate binds to the enzyme less frequently. The reaction converting substrate to product lasts longer, and the random collisions that dislodge the product from the substrate occur less often with less force. Very high temperatures can also slow the rate of an enzyme catalyst reaction. There are many different substances that are secreted into different segments of the digestive tract. Mucus, bile salts, bilirubin, HCl, and sodium bicarbonate are just some of the substances mixed with the foods as it passes through the digestive tract. The small intestine. The duodenum is the first and shortest segment of the small intestine. It receives particularly digestive food known as the chyme from the stomach and plays a vital role in the chemical digestion of chyme in preparation for absorption in the small intestine. Many chemical secretions from the pancreas, liver, and gallbladder mix with the chyme in the duodenum to facilitate chemical digestion. Chemical digestion in the small intestine relies on the activities of three accessory digestive organs: the liver, pancreas, and gallbladder. The digestive role of the liver is to produce bile and export it to the duodenum. The gallbladder primarily stores, concentrates, and releases bile. Insulin is a pancreatic endocrine hormone that are secreted from the pancreas. The pancreas produces insulin and glucagon in response to the levels of glucose in the bloodstream. In order to maintain homeostasis, the beta cells in the pancreas produce high levels of insulin. The small intestine is the site where most of the nutrients from ingested food are absorbed. The epithelial cells of the villi transport nutrients from the lumen of the intestine into the capillaries and lipids. The large intestine. The large intestine absorbs water from the remaining indigestible food matter and compacts feces prior to defecation. On the surface of the large intestine, 3 bands of longitudinal muscle fibers called duodenal coli, each about 0.2 inches wide, can be identified. They start at the base of the appendix and extend from the cecum to the rectum. Both the small intestine and the large intestine have goblet cells, but they are abundant in the large intestine. About 99% of the large intestine and feces flora are made up of obligate anaerobes. And last, here is a general appearance.